Dear team, welcome to the virtual training session. I hope you and your family are safe. Myself Manish from the product management team, and I will be taking you through the training session of our beloved brand ProGG. In this video, we will be covering basics of probiotic, the USPs of LGG, key clinical trials of LGG, the marketing strategy of ProGG, and the action plan. The video shared by our training team gave us a lot of in-depth insights on probiotics. Before starting our training session, I would like to run you through some basics of probiotics. The word probiotic is a union of two words. Pro means for and biotic means life. So if we join those two words together, probiotic as a whole means for life. WHO defines probiotics as live microorganisms which, when administered in adequate amounts, confer a health benefits to the host. The major role of probiotics is to correct dysbiosis or prevent overgrowth of pathogens. Dysbiosis refers to imbalance in microflora population of or within the body and most prominently the gut. Dysbiosis is associated with the loss of beneficial bacteria or the good bacteria, the loss of bacterial diversity, and overgrowth of pathogens or bad bacteria. Dysbiosis is the underlying cause of multiple diseases. We can see the pictures of famous celebrities on our screen. They are considered as the GOAT or the greatest of all time because of certain qualities, and these qualities are being original. They have their unique style which is different from their competitors, and this style makes them original. Example, Charlie Chaplin is known by his small mustache and his unique walking style. Recognized and recommended. We all know who these celebrities are and which industry or sports they represent. If we were given a task to choose the best boxer, the best footballer, and the best comedian, I believe we all would have chosen them over their competitors. They outperform their competitors in all important aspects like talent, skill set, and work ethic. This is the reason which makes them the greatest of all time. Similarly, LGG is a GOAT probiotic or greatest probiotic of all time because LGG is the only original L-rhamnosus GG under the technical tie-up with Christian Hansen, Denmark. LGG is the only Pila-abled L-rhamnosus GG with a better adhesion and clinical efficacy. LGG is genetically stable. LGG, being the original L-rhamnosus GG, is backed by more than 1300 publications. It is recognized and recommended by global healthcare bodies and journals. LGG is clinically proven in Indian patients. Because of these 6 reasons, LGG, the original L-rhamnosus GG from Christian Hansen, Denmark, is considered as the greatest probiotic of all time. Let's understand in detail the qualities which make LGG the greatest probiotic of all time. LGG has fully identified genus, species, and strain. It is a gram-positive probiotic. It was extracted from fecal sample of healthy adult. The strain was named GG in the honor of the scientists who have discovered it. They are Sherwood Gorbach and Barry Goldin, and they discovered L-rhamnosus GG in the year 1985. Pili are like hands of probiotic. They are the most important and sensitive part of the probiotic. Pili governs the clinical efficacy of the probiotic. Loss of pili can happen mainly because of two reasons. First one is the genetic mutation. Secondly, the damage caused during formulation when the force exceeds 8,000 gram. The below mentioned images are of two different L-rhamnosus GG. On the left hand side, we can see the image of LGG, which is the only L-rhamnosus GG with consistent pili expression. Whereas on the right hand side, we can see the image of generic L-rhamnosus GG strain without pili. Pili helps in addition to the intestinal mucosa. LGG, the original L-rhamnosus GG, has SPA-CBA gene. This gene enables the pili to attain a specific conformation. On the left hand side, we can see the structure of pili of LGG. It is made up of three monomers, namely SPA-A, which forms the backbone of pili, SPA-B, which serves as a molecular switch for pilus termination, SPA-C, which binds to the mucin and collagen of the gut. SPA-CBA pili adheres to the gut by two unique mechanisms of adhesion, which are nanospring and zipper mechanism. The nanospring conformation helps LGG to sustain in the gut for a longer duration of time, whereas the zipper mechanism helps to strengthen the adhesion to the host. Because of the unique mechanism of adhesion and the structure of pili, LGG has a stronger gut adhesion than the generic L-rhamnosus GG and the other lactobacilli species. Hence, we can say that LGG, the original L-rhamnosus GG, has a superior clinical efficacy than the generic L-rhamnosus GG. LGG has a presence of a unique biofilm. This biofilm aids mucoadhesion. It protects the probiotic from the external environment, and it also protects the probiotic from the antibiotics which are co-prescribed with them. Genotype means presence of complete set of gene sequence responsible for functionality and its health benefit. When we talk about probiotics, we cannot consider the original and the generic versions as equals, because probiotics are live bacteria, and same probiotics manufactured by different process yields probiotics with different genome or at times faulty genome, and this different genome can affect the clinical efficacy of the probiotic. The generic L-rhamnosus GG strains have a different genotype than the original LGG, and hence that is the reason why LGG shows a superior clinical efficacy than the generic L-rhamnosus GG. Genomic stability of generic L-rhamnosus GG was compared with the original LGG. In this analysis, three isolates of generic L-rhamnosus GG and original LGG were taken. Genetic mutations were observed and SPA-CBA gene was found to be missing in 2 out of 3 isolates of generic L-rhamnosus GG. Because of this reason, the generic strains have lower tolerance in GIT with a lesser adhesion and thus reduced functionality. Antimicrobial activity of LGG is due to the production of lactic acid and bacteriocin. These two agents enter the cell wall of pathogen and lowers the intracellular pH of the pathogen, thus leading to the cell death. Bacteriocin produced by LGG was found to inhibit the growth of several diarrheal causative agents like Clostridium species, Salmonella species, Bacteroides species, E. coli, and Staphylococcus species. LGG secretes proteins MSP1/P75 and MSP2/P40. These proteins inhibit the cytokine-induced IEC apoptosis, and it promotes the growth of intestinal epithelial cell and thus helps maintain IEC integrity and function. LGG produces SCFA, that is short chain fatty acids like propionates, acetate and butyrates from the carbohydrates. These SCFAs serves as a primary source of energy for maintenance of tight junction barrier of the intestinal epithelial cell and thus helps maintaining the IEC integrity and function. LGG is known for its immunomodulatory effect. It releases LTA, that is lipoticoic acid, which along with SPA-CBA pili and the DNA fragment of LGG binds to the toll-like receptors of the intestine and resulting in T cell and B cell activation. T cell activation releases cytokines like IL-10 and interferon beta. These cytokines create anti-inflammatory environment. B cell activation leads to IgA production and release. And in this way, LGG modulates the immunity of the patient. AB blend enhances the colonization of LGG in the gut. AB blend contains two probiotic strains which are Lactobacillus acidophilus LA5 and Bifidobacterium animalis BB12. Both these strains are under the technical tie-up with Christian and Hansen, Denmark. LGG, along with AB blend, betters the intestinal function and augments the immunity. LGG is backed by a plethora of clinical trials. Let's discuss some of the important clinical trials. LGG is clinically proven to reduce the risk of antibiotic associated diarrhea in children and adults by 50%. LGG helps boost immunity by increasing IgA levels by 79% and IgG levels by 26%, which is useful in decreasing repeated episodes of rotaviral diarrhea. LGG shows superior clinical efficacy than Saccharomyces boulardii, Bacillus clausii, and ORS alone in reducing duration of diarrhea, improving stool consistency, and reducing number of hospitalization and fever in children. LGG significantly reduces the incidence of antibiotic associated diarrhea by 72%. A combination of LGG and AB blend is effective in reducing the incidence of antibiotic associated diarrhea in children. Recommendation is given to someone who is trustworthy and reliable. LGG, being the original L-rhamnosus GG, is both trustworthy and reliable, and that is the reason why it is recommended by the best healthcare bodies around the globe. American Academy of Pediatrics recommends LGG for antibiotic associated diarrhea, and it is also recommended by European Pediatric Association and Canadian Pediatric Association. ESPGHAN recommends LGG for acute gastroenteritis. It is also recommended by NASPGHAN and WGO. Apart from the recommendations by global healthcare bodies, LGG is also recommended by the best international textbooks. Harrison's Internal Medicine mentions LGG in the treatment of Clostridium difficile associated diarrhea. Nelson's Book of Pediatrics, which is considered as the Bible of pediatricians, mentions LGG for infectious persistent diarrhea. LGG, which is the only original L-rhamnosus GG, and that is the reason why it is the mark of originality, which is available as ProGG 5 billion live cells. ProGG is recommended in secretory diarrhea, antibiotic associated diarrhea, and immune health. Let's discuss the role of LGG in these indications. The major causative agents for secretory diarrhea are Salmonella species, Clostridium species, and E. coli. Bacteria enters the host through contaminated food or water, which results in dysbiosis. After that, these bacteria enter the intestinal epithelial to release the toxin. These toxins cause damage to the epithelial cell and thus causing diarrhea and the loss of water along with essential nutrients. Antibiotic associated diarrhea results from an imbalance in the gut microbiota caused by the antibiotic therapy. Antibiotics decrease the bacterial diversity in the gut, which results in dysbiosis. Due to it, the pathogens like Clostridium difficile and E. coli colonize in the gut. These pathogens produce toxins and cause damage to the intestinal cells and thus results in antibiotic associated diarrhea. Yes, you guessed it right. This is a scene from the famous movie Diwar, where Amitabh Bachchan says, मेरे पास बंगला है, गाड़ी है, बैंक बैलेंस है, तुम्हारे पास क्या है? To which Shashi Kapoor says, मेरे पास माँ है. Nowadays, the market scenario is also the same, where the companies with generic L-rhamnosus GG are claiming a lot of things, like their strain is original, it is clinically proven, it is backed by international bodies, but only our team can proudly say हमारे पास LGG है. That is, we have LGG, which is the only original L-rhamnosus GG available in the market. Let's discuss the marketing strategy of ProGG. If we look closely at the images, you will observe that they are not the original celebrities. Instead, they are their lookalikes, or in our pharmaceutical slang, we call them as generic versions of the original. Lookalikes can dress like original, they can talk like original, but they are not original. No matter what they do, they cannot replace the original. Therefore, there can be only one original, whereas all the other alternatives are just lookalikes or generic versions of original. Similar is the case in our L-rhamnosus GG market, which is flooded with generic players, but they lack one crucial quality, that is, they are not original. This is the reason why ProGG containing original LGG prevails over its generic counterparts. Let's talk about the reasons why originality prevails over the generic. Original LGG is under the technical tie-up with Christian and Hansen and has LGG logo, whereas generic GG has logo other than Christian and Hansen. In original LGG, SPA CBA pili remains intact, whereas in generic GG, pili may be absent or lost due to microencapsulation. Original LGG has better gut adhesion due to pili SPA CBA, thus better clinical efficacy, whereas generic GG has reduced gut adhesion due to the loss of pili, thus reduced clinical efficacy. Original LGG has stable genome, whereas genomic stability is not guaranteed in generic GG. Original LGG is free from virulent and resistant genes. Virulent and resistant genes may be present in generic GG. Original LGG is globally recommended, whereas generic GG is not recommended. More than 85% clinical trials have used LGG, which is the original L-rhamnosus GG, whereas generic GG has no clinical evidence. LGG, the original L-rhamnosus GG, is clinically superior than the other treatment measures. LGG has 33% better efficacy than Bacillus clozi, 32% better efficacy than ORS alone, and 25% better efficacy than Saccharomyces boulardii. Let's take a look at the competitive scenario of L-rhamnosus GG market. Direct competitors are those brands which have generic L-rhamnosus GG in it. Amongst the direct competitors, Cipla's Unobiotics have a greater chunk of the market, followed by Sundeota's Superflora GG. Bifelac GG and Visilac GG are quickly gaining traction in the market. Indirect competitors are those brands which have probiotic other than LGG. To increase the sales, it is important to keep tabs on both direct and indirect competitors, which can be achieved by a proper and regular RCPA. Success of a brand not only depends upon its value proposition, but also depends upon how this value proposition is being communicated. Let's discuss the 3 C's for successful communication. Clarity. Communication with absolute clarity will boost our productivity, and this clarity will come by regular RCPA, which will give us an insights on prescription habit of our target customers, which would help us to tailor our detailing talk. Conviction. Greater conviction ensures better chances of converting customers because a good message delivered with a great conviction is bound to make an impact. Consistency. Consistent effort is one of the key ingredients for success. A clear message delivered with great conviction on consistent basis is bound to create an impact in the marketplace. In this video, we have learned different aspects of LGG in detail. To yield results, it is important that these learnings are implemented in the right way. Let's discuss the plan of action which will help us to execute our learnings in the right way. The action plan can be divided into three parts: identify, convey, and engage. Identify the top prescribers of both direct and indirect competitors. A regular and proper RCPA will help us to identify our core target. Convey or communicate the product and scientific knowledge which we have learnt in this video to the best of our capability. And lastly, engage the core doctors to build our prescriber base. I hope the details covered by me in this video has served its purpose of value addition. Thank you for sparing your valuable time.